S-acylation is a post-translational modification in which a fatty acid is added to a cysteine residue via a thioester bond. The addition of this hydrophobic element is thought to alter the way proteins interact with membranes and is abundantly present in all eukaryotes. Being first described in the influenza virus in 1979, the S-acylation field was only brought to prominence in 2002 when S-acylating enzymes were discovered. It is still an emerging research field with only 3 laboratories working on mammalian S-acylation in the UK at the moment. We had a closer look into the research of the only lab worldwide working on S-acylation in plants, led by Dr. Piers Hemsley and based in the James Hutton Institute in Dundee. He and his group recently demonstrated that about 1,000 proteins Arabidopsis are S-acylated, resulting in a proportion of 40% of all membrane proteins being modified. The characterizing difference to other lipid-based protein modifications is marked by its unique reversibility, which allows it not only to act as a static structural component, but also as a dynamic regulatory element. Last year, in collaboration with other research labs, the Hemsley group published a significant paper in Nature, reviewing the finding that the cellulose synthase complex is the most heavily S-acylated protein described so far. Moving through the cell membrane, the SES generates long polysaccharide chains of beta 1 to 4 linked glucose molecules, which are then subsequently assembled into cellulose microfibrils. Cellulose stabilizes plants to withstand gravity and grow upright. S-acylation happens mainly in disordered regions of the protein, making these less soluble and more affine to membranes. Blocking S-acylation of the CSC inhibits its insertion into the membrane, and thus the generation of cellulose. Dynamic S-acylation is exhibited in receptor-like kinases, which are the main receivers of extracellular stimuli, thus being part of key signaling mechanisms in plants, controlling its development, pathogen resistance, and hormone signaling. Once bound to a stimulus, RLKs undergo endocytosis and signal from vesicles. It was observed that they are extensively S-acylated in their native state, and S-acylation levels increase upon receptor ligand binding. The addition of fatty acids changes the biophysical properties of a protein, and it was suggested that this alteration leads to either a change in the membrane microdomain composition or to the protein moving from one microdomain to another. Endocytic sites are enriched by certain lipid species, particularly long-chain saturated fatty acids like the ones added by S-acylation, hinting that S-acylation might play a major role in enabling endocytosis and vesicle formation, which is crucial for RLK function. Being able to dynamically manipulate the RLKs by changing how S-acylation acts on the receptors would allow selecting for beneficial traits during agricultural processes, such as improving the pathogen response. The 2 major next steps in understanding the potential role of S-acylation in regulating membrane protein functions are identifying which proteins are S-acylated and how protein S-acylation is regulated. The Hemsley group is currently carrying out proteomic studies to specify which proteins are S-acylated. As for the regulatory mechanisms, so far 24 protein S-acyl transferases have been described in humans and 25 in plants. The PAT family are integral membrane proteins, with each PAT being found only in a subset of intracellular membrane compartments. PATs therefore are thought to gain their specificity mainly from spatial separation, meaning that the trafficking route of the protein determines its S-acylation state. In contrast to mammalian PATs, most plant PATs are located in the plasma membrane, and only a small number resides in the Golgi and the ER, hinting that the proteins are targeted to the membrane on their own, before being trapped in it by S-acylation.
The focus at the moment in the lab is understanding how S-acylation affects plant-pathogen interactions, particularly the dynamics of S-acylation. What we're speculating is that if we can manipulate S-acylation in the receptor, we can either change the speed of the response, the magnitude of the response, or the duration. So by manipulating the receptors, we might be able to actually get a receptor that activates in response to pathogens much more rapidly and has a much longer response, but isn't quite as strong.
However, little is known about the way plant proteins become de-S-acylated.
We're trying to identify the de-acylating enzymes, partly so we have tools to work with. So if we understand which enzyme's doing what, we can understand is there any feature of a protein that acts as a recognition motif for recruitment of the de-acylating enzymes. So we can understand, can we manipulate so that we can slow down the de-acylation reaction. It also gives us the tools to actually understand why and how this happens.